I want to start by thanking all of you for staying engaged with the materials in this course. Today, we're moving into our final module, Module 7, which focuses on seed viability and viability testing. In this module, we'll define what seed viability means, discuss why it's important to test for viability, and explore the different methods used to evaluate whether seeds are still capable of germinating. By the end of this section, you should have a clear understanding of both the concept of seed viability and the practical tools that scientists use to measure it. Seed viability can be understood in different ways depending on the context. In the seed industry and among seed analysts, viability is defined as the ability of seeds to germinate and develop into normal seedlings. In this sense, viability is essentially the same as germination capacity. It is important to note that viability is not usually considered at the level of a single seed, but rather at the level of a seed lot, where the population as a whole demonstrates a certain level of viability. For seed scientists, the concept of viability goes beyond whether a seed can germinate. Viability refers to the degree to which a seed is alive and metabolically active, with enzymes capable of driving the biochemical reactions needed for germination and seedling growth. Under this definition, a seed may contain both living and dead tissues, and it may or may not be able to germinate successfully. In other words, seed scientists consider both the viability of individual tissues within the seed and the overall viability of the seed as a whole. Seed viability is highest at physiological maturity, the stage when the seed has fully developed on the parent plant. From this point onward, viability gradually declines as the seed approaches harvest maturity. Recall that physiological maturity marks the end of dry weight accumulation in the seed and is characterized by the detachment of the funiculus from the ovary wall. At this stage, seeds typically contain 20 to 50% moisture content, depending on the species. In contrast, at harvest maturity, seeds have already undergone a controlled process of desiccation, reducing their moisture content to about 10 to 15%. This lower water content slows metabolic activity, allowing seeds to remain viable for extended periods of dormancy or quiescence. Although seeds at harvest maturity are alive and capable of germination, the environment within the parent plant prevents them from sprouting prematurely. Knowing that viability declines as seeds move toward harvest maturity, the next question is, how do we measure whether seeds are still capable of germination? This brings us to the importance of seed viability testing. If someone asks, why do we need to test seed viability? The intuitive answer would be that we want to know how seeds will perform in the field. However, viability testing is not designed to mimic field conditions. Instead, it is an analytical procedure that evaluates whether seeds are capable of germination under standardized, highly favorable conditions, conditions that seeds rarely encounter in the field. Because of this, viability tests often overestimate the actual field performance of seed lots. This naturally raises the question, if viability testing does not perfectly predict field performance, why do we still rely on it? Even though viability testing may not perfectly predict field performance, it remains essential because it provides a consistent, standardized baseline for evaluating seed quality. In the seed industry, the use of uniform procedures across laboratories ensures that comparable germination percentages can be obtained from the same seed lot. This consistency also allows verification of germination results printed on seed labels, ensuring accuracy and transparency for buyers. For seed producers, viability testing makes it possible to compare the quality of different seed lots before planting decisions are made. Finally, the information gained from these tests help determine both the economic value of a seed lot and the storage requirements to maintain its quality over time. For seed banks, viability testing is also of primary importance. Seed banks are specialized facilities that conserve plant genetic resources by storing seeds under carefully controlled conditions. Their primary purpose is to safeguard biodiversity, protect against genetic erosion, and ensure that valuable crop varieties and wild relatives remain available for future use in research, breeding, and conservation. By maintaining these collections, seed banks act as living libraries of genetic diversity, providing a critical resource for global food security and ecological resilience. Take the International Rice Research Institute, or IRI, in the Philippines as an example. IRI maintains more than 140,000 rice successions, including land races and wild species. To protect the integrity of this collection, seed viability must be monitored regularly in both the base and active collections. Seeds in the base collection are stored long term, typically for 15 to 20 years, while seeds in the active collection are maintained for shorter periods of about 5 years. Routine viability testing is essential in both cases. It ensures that seeds in long term storage remain alive and that seeds distributed from the active collection to research partners will germinate successfully. In this way, viability testing guarantees that the material provided is both living and usable, supporting global research and conservation efforts. This slide illustrates some of the practical aspects of seed viability testing carried out at IRI. In the leftmost picture, a technician is preparing rice seeds for germination testing using the moist paper towel method. The middle picture shows rice seeds already germinating in moist paper towels placed inside an incubator, where temperature and humidity are carefully controlled. In the rightmost picture, rice seeds are being germinated in petri dishes lined with moist paper towels, another common setup for monitoring germination. These images highlight the standardized procedures used to evaluate seed viability, ensuring that results are consistent and reliable across tests. We've just looked at how viability testing is carried out in practice at IRI, where seeds are actively monitored and distributed for research. But seed conservation isn't only about day to day use, it also requires long term safeguarding of genetic resources on a global scale. That brings us to one of the most remarkable facilities in the world, the Svalbard Global Seed Vault. The Svalbard Global Seed Vault, located on the Arctic archipelago of Svalbard in Norway, is the world's largest secure seed storage facility. Often called the Doomsday Vault, it serves as a global backup for gene banks around the world. Seeds are stored deep inside a mountain under permafrost and rock, providing natural refrigeration and protection against natural disasters, war, or equipment failure in national seed banks. The vault currently safeguards over 1 million seed samples, representing thousands of crop species from nearly every country. Its purpose is not day to day distribution, but long term preservation of genetic diversity to ensure that in the event of loss elsewhere, seeds can be retrieved and replanted. Even here, routine viability testing is essential. Seeds stored in the vault must be periodically tested to confirm that they remain alive and capable of germination, ensuring that this global safety net truly functions as intended. In summary, seed viability refers to the ability of seeds to germinate and produce normal seedlings. It also denotes the degree to which a seed is alive and metabolically active. Seeds may contain both living and dead tissues, and not all of the living tissues are necessarily capable of germination. Viability testing adds value to a seed lot by providing critical information about its quality and by determining the appropriate storage requirements for the seeds. Several methods exist for testing seed viability, but the germination test and the tetrazolium test are the most widely accepted and commonly used worldwide. We will be discussing them in more detail in the succeeding lectures.